All right, number one, maybe this is obvious, but maybe not. There is no science to be talked about without the data. So if you are heading to graduate school in the sciences, your ticket to get out of graduate school and go and live a real life is data. And there is actually a certain amount, it's different in each program, a certain number of data figures that you are expected to produce to add to the knowledge of the world before they will let you out. There is no science to be talked about without the data. The data is the science. The data should be central to what you're talking about. So that's point number one. Okay, so part of the time today we're going to do group work. So to get started with what we're going to do next, you need to be in a group with 2 or 3 other people, and at least one person per group needs to be writing down what your answers are to the questions that I'm going to ask about data, real data that I'm showing up here. So make a group. Groups, yes, groups of 3 or 4. You got a group? Ready to write? Okay, so you've got your group. I'm now going to recreate a common moment in science seminar. Are you ready? I'm going to recreate a common moment in science seminar, which is that a figure appears on the screen, and there's some data, and normally someone starts talking about this data after you just saw it 3 seconds ago. So what I want you to do now in your group is to make a list of what you need to know about this to understand what the data is saying. Make a list of things that would help you to understand what this figure is saying. Shelby, you might not want to participate in this, because you already know what the data is saying. So make a list, and I'm going to ask different groups what's on your list. What do you need to know? I should have made all chemistry. Start.
Needs to have a larger
Yeah.
a larger Somebody who's never seen it before. That's right, that's what I asked. Well, I know what my conjugate Yes, what's that? If it was in the air. Like, I think they go read that grass. I feel like I have to do a
Write that down.
I mean, that's literally all you can say about it.
Okay, you probably could keep going for a while, but we're going to stop there. And I'm going to ask groups what one of your questions was. And so I would like for, as I go through successive groups, try not to repeat the same question, because they're probably the same questions. So this group, what was a question or an item that you need to know to understand this graph? What is the stimulus? So we've got something describing a stimulation up here. What is the stimulus? This group, what was one of your questions?
Clarifying the key.
Clarifying the key. What is CN? What's 3%, 7%, 10%? This group.
So is this the complex 1 and 2 on the same graph?
Does this represent complex 1 and 2 on the same graph? That's a question. What about you?
What each color represents?
What each color represents. Next.
I know.
They got all your questions so far?
Pretty much.
This group? I'm just going to point down the rows, and if you've got a unique question, answer it.
Why is it declining?
Why is it declining? Group over there. Explain the X and Y axis. Thank you. Other questions? Who's got a unique question? Yeah.
It could have just been worded different, but what is the significance of the percent as the time increases?
Where do the outliers come from? What's the significance of the percent? What you said?
Yeah, how the O2, what's the significance of
Oh,
this time
what does
increases?
this percent mean? Percent O2. What about back there?
What is the expected result and does it follow the pattern?
What is the expected result and does this follow the pattern? Other? Yeah.
What is this P
Uh,
0.05 mean?
What does this P less than 0.05 mean? Maybe some of you in here, you don't have to raise your hand. What is a mitochondria? What is complex 1 and 2? Depending on your major, I would expect you to know that. There are a lot of questions that need to be answered about every single data figure. So, if you are presenting this semester or any semester, it's really important for you to remember the feeling, remember the feeling of going from this to this. Once you start going into your prepping your presentation and you look at the papers for umpteen hours and you spend time with your advisor on each figure for quite a while, it's easy to forget what it's like to go from this to all of a sudden there's this graph up there and it's discombobulating. Is that a word? It's discombobulating. So what I would suggest to you is that it's not possible to explain data without explaining a bunch of other stuff first. Before you explain data, there's a lot of other stuff that has to be explained first. A lot. So, before you can explain the data, these are some things that you as the presenter have to know yourself? Well, obviously, you have to understand the data figure yourself. If you do not, let me give you some advice. Do not put a data figure up if you are barely grasping what it means. It will become obvious if you are barely grasping what it means. So work with your advisor. We are here to help you understand what it means. I'll come back to that later. You have to understand the data figure yourself before you can explain it to other people. To understand the data figure, you need to understand the method that the researchers use to get the data. Often, that's the hardest part. What did they do to even get that data up there? Because you learn from a very young age, like how to look at a graph and describe a trend and describe the axes, but the methods are what tend to trip students up. So I'm going to talk about ways to deal with that understanding. This is a preview of today, basically. You have to understand what was the statistical analysis, if there was one. What was the statistical analysis that was used to make that data? Why are the researchers deciding to make the data in that form and not a different form? Some of you, when you present, there's going to be a choice of whether to show a table of data, which usually is boring for the audience and hard to know where to look, or a graph of the data, or a picture, a photograph of one particular result. How do you know why they picked that particular data form? You need to know what every word on the data figure means and every abbreviation what it means. And also, this is a pet peeve of mine, is knowing what the data really say and what the data really don't say. What that data figure is saying is something very specific. What does it say and what does it actually not say? So when you're getting ready for seminar, or when you're getting ready to present data at a conference, or when you're getting ready to present data in your class for your presentation, you start with the data and then you build everything else around it. That's not often the way that students would like to do it, because the data is hard and the background is fun. So, yes, the background is fun. The data is the science part. The data is what you should start with, and then work backward from the list of questions to figure out what else you need to say to explain the data, because the data is the science. So I did this myself as a student, totally. I made a presentation on the size of potatoes growing under different fertilizer regimes, because that was an easy paper. I did that myself. I completely understand the desire to pick a paper where it's easy and it's easy to understand the paper, and you don't have to ask for help. That does not help you in the long run. So when I did that, I didn't grow in my understanding. I didn't stretch to learn about new techniques. I know exactly what it feels like, and I did it. I'm telling you, don't limit yourself to that. Don't limit yourself to that. And when you do find a paper that's cool, if you come across a figure that's a key figure and you can't tell what it's really saying, it doesn't mean that you shouldn't use that figure. It means go talk to your advisor, because one reason that we spent umpteen years in school was to training us how to explain to you in 10 minutes what that figure means. That's why we're here. That's why you have Science Seminar Advisors. So ask us. Keep us sharp. So keep in mind, like that feeling you felt when you saw the graph with colors, what is that? Except for you and your advisor, nobody else in here, which is about 60 people, has seen the figure before the minute you put it up on the screen. So even the professors are really quickly scrambling in their mind to figure out the figure, what it says, the minute you put it up on the screen, unless it's your advisor. So what I would like to see in seminar is for students to spend more time explaining data than they have been in the past. And when you're explaining data, you need to assume that everyone else doesn't know anything about it because everyone else doesn't know anything about it. Also, we tend to have more biology students than chemistry and physics and forensic science students. If you're a biology student, you need to be explaining to all the students. If you're a physics student, you need to be explaining to all the students. So I will use the example last year of Ro Avia, who to me had one of the most hard topics I've ever seen in seminar and did everything in his power, I thought, to make it explainable to a general audience. Anybody see that? Well, half of you probably saw that. He really did a lot to bring it down to biology people. I still was barely hanging on. That's okay. I could tell that he was doing things to try to explain it to me at a biology person's level. So remember who your audience is. All right, now here's another figure. This is a real figure. There it is. You just saw it. If you know what this is, probably need all the lights out on this. If you know what it is, don't say it. Don't say it. You can't say it. You're giving it away. These could be blood-sucking leeches full of people's blood. These could be patterns of lava running down the side of a volcano. These could be spirochetes that cause syphilis. Or these could be fluorescently labeled nematodes that are glowing in correlation to how many neurons they have. D is the closest answer. But you don't know just to look at it like this. You've got red glowing things, and you've got numbers, and you've got CN. So how do you go backward from this to be able to describe it to an audience? Okay, so there are several strategies to be able to talk about data. I'm going to go through a list, and then from time to time I'm going to ask you to do the group questioning again. All right, number one, is the data clearly visible? Notice how Dr. Fitz just had to turn off the lights to see that. There are going to be pictures that you may have in your presentation where you will need somebody to turn off the lights so we can see what it is. There are other cases, and you've seen them in here, where somebody blows up a figure that was this little, and they blow it up on the screen, and it's super pixely. There are other cases where somebody puts up a table that's huge, and you can't read what they're trying to show. Part of the reason that it's good to use papers from the last 5 years is that you will have fewer pixely problems if you're using modern papers. I'll show an example of this in a minute. If you're going to use a table that has teensy tiny data and you're only going to talk about part of it, you can't talk about all of it anyway. It's okay to recreate the table with the parts that you really want to talk about. So make sure your data is visible. Here's an example of a data figure that could be more visible than it is. This is from one of my papers in 2006. It's not super blurry, but it could be better. If you end up with a blurry figure, the best thing to do is go back to the paper, try to find high-resolution images. My paper is so old, it has no high-resolution images. I have them on a thumb drive at my house somewhere. But because I was in a hurry, I got it off the internet. It's this blurry, best I can do in this presentation, and hopefully it's visible enough. So here's an example of maybe slightly less good resolution in a figure. So in your group, go through the same process. What do you need to know about this figure to figure out what it means? Go.
She just said that a while Probably says lunch, but it is. Obviously, it is. Yeah, they came in a pack of 8. They were like, and I like different colors. That's, well, I'm really in the thing. That way we can I tried, yeah. My room, not too much. Everything else. All horses look good. But I got so much except
Okay, I'm going to start in the back this time. Back there. What do you need to know to figure out what's happening in these sleeves? What is HPI? That's a good one. Actually, for this one, just raise your hands and say stuff. What else do you need to know?
What is the title?
What is the title? So, to me, I don't prescribe that to my students that every graph has to have a title if the student chooses for it to not have a title. Titles are helpful if you can come up with a good title that won't distract people from looking at their graph. That's a good one. To me, as an advisor, it's a choice whether students put titles or not. Good comment. What else?
What is the significance of the color differences?
Why are some parts of the leaves not green? To paraphrase what you said. That's pretty important. What else do you need to know about this? Yes?
What kind of leaves are they?
What kind of leaves are they? They happen to be soybean leaves. That's important. Yes?
What are the letters and numbers on the pictures?
What are the letters and numbers on the pictures, which are, there's a lot of them. Yes. Yeah. I'm not going to go into all the details of this because it's not the point, but what are some other questions about this? Yeah.
Is this one leaf or multiple?
Good question. Is this one leaf or is it multiple leaves? They happen to be multiple soybean leaves containing bacteria with multiple different mutations in a certain protein. If the leaf turns brown, it means that section died. If it died, it means that the leaf can recognize the bacteria, kill itself off to prevent the bacteria from spreading to other parts of the leaf. I would have to do a lot more work beforehand to make that more understandable. So those are the short answers to the questions. But if I were going to present this, which I did many times, PhD research and at posters and so on, I have to anticipate what the questions are and prep people beforehand to know that D297A is a mutation in the aspartic acid at position 297 in the bacterial protein that turns it into an alanine. Blah, 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 blah. It takes a long time. That's part of the point. So to explain the data, there's a lot of stuff behind it that has to be explained. All right, abbreviations. This picture I picked because it has too many abbreviations. This is a lot of abbreviations. And most of you, when you present, you're going to have a lot of abbreviations. Science journal articles, you get a certain amount of room, you abbreviate as much stuff as possible, but when you are not in the field, you're not going to know what all the abbreviations are. What's WT? That's wild type. If you're not a bio major, you can't assume that a physics major knows wild type. Thank you for shaking your head no. And proving my point. This one happens to be empty vector. Then you've got all these others. HPI is hours post inoculation. So how do you deal with abbreviations? I have 3 suggestions. The first, I have seen people attempt, but have not seen it done well yet, which is to write the most prevalent abbreviations on the board legibly. If you were in here previously, you've seen people try to do this. This would be the correct way to do it, which is you write really big and you leave enough time that you're not trying to do it 30 seconds before your talk. HPI is hours post inoculation. A lot of times people write it right here and then they stand in front of it and you can't read it anyway. This is a way to deal with that. Second way to deal with that is you could, I haven't seen anybody do this, but you could make handouts and bring them in for everybody. Or you could put like a little key on certain figures to where wild type, EV, empty vector, HPI, hours post inoculation, and then these individual mutants. If there's an abbreviation that you're going to be using all the time, or if there's a top 5 that you can make some type of help for your audience to figure out what they are, that's really good. Number 3. This is the last practice figure. So in your groups, discuss what do you need to know about this figure, which requires the lights to be off, thank you. What do you need to know to understand this figure? This is a stain. What do you need to know about this? Yeah. Is this a progressive sample? You mean over time? Title would be good on this one. What else do you need to know about this?
Is there something that's added to it and taken away?
Is something being added and taken away?
What kind of staining method?
What kind of staining method? I heard somebody say, why are they green or not? Yeah. What's the measurement?
What is it?
Hmm?
Like what it is? There's nothing.
Yeah. What are the blobs? Actually, in these, there's a slightly yellowish blob on a white background.
Yeah. No, I mean,
Yeah.
I meant like there's
Yeah,
nothing
what
there
is it?
explaining what it is.
What is this? Okay, this is an example of a particular method in molecular biology called yeast-2 hybrid. And this relates to, if you're going to show data like this, which this actually was really important information, if you take my word for it. I would have to provide a lot of information about what that was, because that's even more mysterious than the leaves. Like the leaves, obviously, some parts of the leaves are sick and some parts are okay and green. This one's really weird. This is an example of a technique called yeast-2 hybrid assay. Normally, if you heard about this in undergraduate class, it would be in molecular biology, but you might not even then. So, in this case, if I had a student that needed to show yeast-2 hybrid data, as an advisor, I would expect that I would explain to them how it worked. Same for your other advisors. If you have, I'm going to use Keisha as an example. Keisha has, one of her figures is using optical dichroism. Is that right? That's it, right? See, I told you we were excited. So, optical dichroism is something that I can explain decently well. Dr. Fitz is obviously excited by it. Either of the chemistry professors could also explain it. Any of us in 5 minutes or less. So if you come across in your paper a figure that seems to be important and it's optical dichroism and you don't know what that is, go to your advisor. Oh, Chip also knows. No? You could probably figure it out. Just based on the root words. So, first, go to your advisor, ask them about the method. If we don't know, we'll get back to you the next day. We dorkily get excited about methods that we don't know about yet. Trust me. That's why we're here. And we can help explain to you the method. You shouldn't feel like a bad student or a dumb student if you don't know what the method is talking about. Because part of the process of Science Seminar is to get you to read papers and reach and struggle with terminology and methods and so on. That's part of it. Because once you've done it once, then you're not so intimidated the next time, and you know how to figure out methods.
But don't wait too late.
Oh yes. Okay, so over there, a minute ago we were talking about a student last year who had a method that none of us were familiar with that asked us 2 days before. 2 days before. That's not enough time. So in the syllabus, when it says you should meet with your advisor 2 weeks before to go through your slides, that's the time. Really, as soon as you read your paper and decide on your topic, you should figure out the figures that you're going to use. That's the time. Because even if we can explain it in 5 minutes, you asked us the day before, that doesn't mean that you're going to be prepared to talk about it at seminar. So complicated methods are not a reason to avoid picking a certain topic or a certain paper for seminar. We expect you normally to have to struggle with the methods. All right, so this method, yeast 2 hybrid, it so happens that this is a fairly standard technique, and you can go and find diagrams that help explain it on the internet. So I Google imaged, I found this. This is the link. Because this didn't come out of one of my major papers, I'm citing it. And also because I didn't draw it myself. If you draw it yourself, or if it comes out of your major papers that you're talking about, you don't have to cite the link. But this technique works. What it does is it's a cheap way to figure out if two proteins interact and bind to each other. In this example, I have a protein called RIN4. I have a protein called ABRB and its mutant derivatives. I hook up RIN4 to a DNA binding domain using molecular biology techniques. I hook up RIN4 physically fused with the activation domain, and if they touch each other, if they bind together, these two are close enough together that they turn on expression of a blue protein in yeast. Super helpful. The blobs are yeast. The bluer they are, the stronger the interaction between RIN4 and ABRB. So the bluer it is, the tighter the binding. The numbers measure blueness. This is a complicated figure. It doesn't take that long to explain if you know what you're talking about. Get your advisor to help you know what you're talking about. All right, so is your data visible? Did you talk enough about the methods? And this is one of my favorite. Did you rush through the data? I haven't discussed this with other professors, but I almost have an internal clock in my mind when I'm watching you present your data, and if you jump off of a data figure in less than 30 seconds, I have an alarm going off in my brain. It's like, oh, they skipped over that data. Make sure that you're not rushing through the data. So back to the lava, or the leeches, or the C. elegans with labeled nervous system. Thank you. So there was this figure, and to explain this figure, you would need to know that each one of these is an individual worm out of a group of many worms that were treated under various conditions. These worms were treated as a negative control. This group of worms, which was treated with 3% of a certain pesticide. This group of worms was treated with 7% of a certain pesticide, and this group with 10%. And the amount of glowiness of the worms correlates with basically how many or how much neuron material they have. Is that correct?
That's good enough for the purposes.
Yeah, okay, thank you. Thanks for the graciousness. So here's 4 worms, and they're showing different levels of glowiness. That's pretty nice. But to really make this mean something, these are representative worms. You need the graph, in this case, so that we're not just talking about 4 worms, we're talking about 4 groups of worms. The negative control had this much fluorescence, which represents this amount of glowing neurons. The 3% pesticide group had this amount overall of glowing neurons. 7% group, 10% group. A lot of times students will tend to show this data and then get asked in the follow-up question about the quantification, the numbers behind it. If you have time, go ahead and show these are like sister figures. If you've got time, show the quantification. If you don't have time, you need to have this for question time later, because we're going to ask you. Because this on its own is not, anything. No offense to whoever made these pictures, but these are representatives of the numerical data. All right. Another question. Did I address this? This is a word I struggle with. Did I address the statistics associated with the data. I'm going back to that same figure I just had. Another common thing that students tend to do is they will show a graph, and hopefully they talk about what the axes mean. Hopefully they talk about what the height of the bars mean, and then they go on and ignore the error bars and the statistical significance asterisks, which is another hard word to say. If you do that, we're going to ask you later. If you rush for time, maybe you need to skip over it and wait for us to ask you later. But normally we would expect you to address what do these error bars mean, and what does it mean that these two have 3 asterisks and these two have no asterisks, or stars. I'd be okay if you call them stars. Obviously. And normally, the convention is that when there's 3 asterisks, stars, it would mean that this column is different from all 3 of the others, statistically speaking. Is that correct in this? No?
In this case, the asterisks mean that those that have them are different from the control,
From the control.
just from the control. That is in the
This-
figure legend, though. That would be in the figure legend. It may not be in the body of the article, but that information is almost always in the figure legend.
True. However, the figure legends that are in the paper are not always appropriate to put on here for your seminar presentation, because people are going to read them and get tripped up by certain stuff in the figure legend that's too much information.
Please don't put the figure legend on there.
Because they're like paragraphs, usually. But you do need to address the stars, address the bars. Stars and bars. All right, another one that's a pet peeve of mine is putting up a data figure. Maybe even if you did everything perfect and then you say something about the data figure before you click on that is a lie about that particular figure, because you're saying more than the figure says. So example, well, I have an example of overstating what the data says. So back to this. You might say, after showing this set of figures which go together, that this data shows that glyphosate is poisonous to animal nervous systems. No, it doesn't. That's not what it says. Even if the title of the paper happened to be glyphosate is poisonous to animal nervous systems, that is not what this says on its own. It is not. And if you were a sharp observer of seminar in the past, you might notice, first of all, you might notice that the number one question is, could you go back to figure X? We hardly ever ask you to go back to a non-data figure. You ever notice that? Could you go back to figure X? Because the data is the science. And so if I heard you say, based on this, that glyphosate is poisonous to animal nervous systems, I would raise my hand and ask you to go back to figure X and ask you to explain why this shows that, which is a leading question because it doesn't show that. So this is in all science, whether you write, whether you present data, whether you're talking to your friend about global warming, whatever. Don't overstate the data. Don't overstate the data. It's not what that says. This is a better explanation. It's not the best explanation because this is not my data figure, but a better explanation is that this data shows that intermediate levels of glyphosate, these two, alter neurons in C elements. That's all this shows. Maybe it doesn't even show that much, but that's a better explanation of what this shows. So what your paper claims in the title is the result of all the figures. Don't use the title claim on a figure. It's overstating. Okay, so these are things to keep in mind when you're preparing for science seminar, especially. Class presentations also. Other times when you're talking about data elsewhere. In seminar, you need to have 3 to 8 figures that come from your primary research papers. You're supposed to be talking about at least 2 research papers. You need at least 3 figures. That's really on the low end. If you're talking about 3 figures, they better be really hard to explain figures, and sometimes that happens. If you're going to fit in 8, a bunch of them need to have the same methods. So Tyler Gavey last semester was talking about, it wasn't that long ago, but he had a bunch of bar graphs that had all the same methods, and once he explained the method, he can just keep putting up another bar graph, another bar graph, another bar graph. He, out of my advisees, he had the most figures because they were easy to talk about. Don't plan on putting 8 unless half of them have the same methods. So the first trick is to identify what are the key figures. A lot of times it's the early figures in the paper where they make like the really boom claim, and then they go on and do other experiments to flesh out details. So the number you pick depends on how hard they are to explain. If they're really easy to explain, you need to go find more figures. You start with the figures, and then you work backward. Those of you who are really passionate about your topic, I know how much you cherish the background information. Cancer is a popular topic where we talk about how many people get cancer and how many people die from cancer. That's almost common knowledge. That may not be where you need to spend your time. Work backward from the figures, figure out where you need to spend your time. So you make a list like the one you made in your group. What do I need to explain this figure? You might have 3 lists. You might have more than 3 lists if your figures are easy to explain. This is where the time comes from. 20 to 30 minutes. It really should take close to 30. I know you tend to speed up because you get nervous, but even so, it takes time to explain data well. Work backward from the data, figure out what you need to say about methods, about statistics, about abbreviations. The background fits in around everything else. The conclusions fit in around everything else. That's the way to do it. It takes time, it takes work, but if you can do this, this is an amazing life skill. Start with what's hard, work backward, figure out what you need to say to your class, to your colleagues, to your boss at the big presentation, on your job interview. Really good life skill. And my last advice is to work with your advisor early and often. I limit how many students I advise in a semester because I expect to be spending multiple hours with you working on this. All of the advisors would feel the same. Work with your advisor. We're here to help make this less painful and more educational, and for you to do a great talk and get these skills under your belt. Come see us. We love it. We love it. That's why we're here. So I have certain announcements, don't leave yet. Number 1, you need to sign in on the sign-in sheet before you leave. If you were absent last week, that was your one freebie. If anybody's been absent 2 weeks, they're going to have to make this up. Announcement number 2. Where'd it go? There's going to be a talk next week at Emory and Henry College about one of the most important It's by one of the most important experts in monarch butterfly studies. This guy right here, Dr. Lincoln Brower. He's giving a public lecture on monarch butterflies. If you're interested in that topic or topics related to insects or ecology, this is a great free lecture that you could attend next Wednesday night. And it's free. And if you're a fan of Barbara Kingsolver, who's quite a renowned author, she's going to be there too. That's announcement number 1. Announcement number 2, this is the absolute last moment, hour to sign up for the biology picnic if you have not already done that. There's going to be several people there. It's going to be fun. It's Thursday at a beautiful farm. It's not far away. It's early in the semester. You have time. It's a great time. Sign up after seminar if you want to attend so that we can get food for you. Other announcements?
2 weeks from today, we have some visitors coming from Eastman Chemical Company, including one of our alums. There will be opportunities for a few of you to go to dinner afterwards. Let me know if you have a particular interest in doing that with Nikki Starnes, who's an analytical chemist, and Jason, who was a student here earlier. So
Who was the
there's an
student?
opportunity. It's 2 weeks. Yeah.
Who was the student?
Hold on, I'm thinking, which could, I'll have to check and see for sure. I think she was going to have, hang on,
You've got
Matt, not Matt,
and Josh Taylor?
Josh Taylor's who's going to be, she was going to check with them. She hasn't gotten back to me.
Okay.
Because Josh is now in a different division. And so I don't know whether Matt is our backup. But Nikki Starnes is a PhD analytical chemist who's been there for a while, and she is our liaison for Technician Highland.
So if you think that you even might someday want to work at Eastman, you need to be at this dinner. It's free.
Well, and I don't know how many we can take, we can deal with that. So
Better
that's why I
tell
said
them.